have a big experiment. I'm going to tell you everything about it, and I'm glad to come just after because we keep talking about diabetes, so we can have different points of view about the potential treatment. Okay, so I will start introducing to you the OptiFlow because this is our experimental setup. Okay, so so what you see in the video is an OptiFlow. The OptiFlow is a field of motion that is produced on the retina when we move to the environment. So basically, we are able to move in the environment just because we move. Otherwise, the world is steady. In this video, you see a driver, and the arrows represent the direction of motion of the single object, single frames in the visual scene. And you see when the driver goes right, the arrows goes left, and vice versa. So we are able to move and navigate in the environment just because we use this relative motion between ourselves and the object in the environment. So optic flow is particularly important for us because we stimulate the subjects. Sorry, I apologize, the wrong button. So we stimulate the subjects with this kind of stimuli in the laboratory. So to give you an idea of what I'm talking about, when we move in the environment, we often experience something like that. So this is a video representing a person moving in a corridor. And this is an experimental stimulus in which the person is in front of the screen, and we simulate passively motion moving dots. So these moving dots for our neurons represent this kind of stimulus. So for the neuronal cells, it doesn't really matter if we move in the environment, like in this case, or if we are standing in front of the screen where it projected a 3D visual stimuli. An important concept.
So just have to change the view. I think they don't see like this patient.
Okay.
I think they see just
maybe it's not shared.
No, I mean I think we have to share this one. Yeah, no, they see. No, they see.
Okay, fantastic.
Okay, cool.
So, okay. So, an important concept when we talk about optic flow and psychophysical study is this point, which is the focus of expansion. The focus of expansion is the specific point that originates motion. Here, you see in two cases. And the focus of expansion is like this only when we fixate the final destination of our motion. So if we go left or right, you don't have this situation, you have an acceleration on the left side or right side. This is particularly important because in our daily life, we very rarely focus our attention and our gaze on the focus of expansion because usually we walk looking somewhere else, not our final destination. And this is very important because we need attention to navigate in the environment. Even if we don't perceive to pay attention to something, we need attention, and these mechanisms seem to be governed by microsaccades. Microsaccades is a very new field of study because we have the instruments to study microsaccades since the beginning of 2000, more or less. people postulated the existence of microsaccades even in the 50s, in the past centuries. But the eye trackers at that time worked with a very low frequency, recording frequency, so it was not possible to detect microsaccades. So what microsaccades are? So you see here is the blue circle is fixation, and then like a moving yellow dot represents the vision of one eye. So this is the behavior of our eyes. So eyes are never still. Even when we fixate an object, eyes are really never still because they always make movements. Movements could be tremor, drifts, or microsaccades. Microsaccades are still a brand new field of studies. So we don't know much about microsaccades. Some people say microsaccades are useless, so they don't study nothing. Somebody else says that microsaccades are useful to avoid fading from perception and to avoid adaptation from receptors. Other studies postulate that microsaccades are useful because they are indicating the unconscious focus of attention. So those are fixational eye movements, basically, but the direction of the microsaccades could indicate where we unconsciously focus attention. And this is particularly interesting for our fusion study, in which we study the mechanisms that allow us to move in the environment. So now we go a little further, going back, let's say, to the diabetic retinopathy. In our lab, we started studying the diabetic retinopathy about 10 years ago. Because diabetic retinopathy from an experimental point of view is a very good model to study perception. Because in this figure you see the difference from an healthy eye and a diabetic retinopathy eye. In diabetic retinopathy situation, when there is this complication, people experiences aneurysms or microaneurysms, hemorrhages or cotton wool spots, and abnormal growth of new blood vessels, so a very large neovascularization within the retina. The issue with the diabetic retinopathy, and also everything I would say related to diabetes, is that diabetic retinopathy is a very slow progress of a diabetic complication. So at the very beginning of diabetic retinopathy stage, people do not perceive any symptom. And this is the worst situation because these people in the early stage of the disease are convinced to have a normal sight, while the ophthalmologist already detect microaneurysms and leaks, But in this very moment, in the beginning of the disease, of the diabetic retinopathy, we can clearly study an eye which is already damaged, but the person feels to have normal perception. So it's very important for us because we can deepen the knowledge of the mechanisms, both cortical and subcortical, and the balance between visual system, postural system, and other systems. So just a little bit of more explanation about the diabetic retinopathy, as we already know from the previous talk, the hyperglycemia causing oxidative stress, causing retinal neovascularization, and this retinal neovascularization, together with the aneurysms and hemorrhages, could cause loss of vision in adults. Of course, all these steps take years, many years. Not everybody lose vision, but it could happen. happen. And anyway, in the later stage of the retinopathy, the vision is impaired. So we need to act in the way that these people really have a better quality of life, even with diabetes. Okay, so our tool to try to avoid the development of both diabetes and its complication is a physical exercise. It seems to be very banal. It's not a medication, but it's known from a lot of papers in literature, and there's a big consensus on this, that the practice of physical exercise reduces the glucose level in the plasma, because the physical activity or physical exercise augments the glucose uptake in skeletal muscles via insulin-independent pathway. Basically, it's the muscle that with the exercise cause more glucose into the cell. So the regular physical activity enhances the beta cell function, the insulin sensitivity, vascular function, and the functions of the gut microbiome. So the physical exercise is a very good way to control the plasma glucose level and help in management of diabetes. Because it's free, you know, and it's medication free. So this is something very easy that everybody can play or take as routine, let's say. I tell you our aim in this very big experiment because diabetic retinopathy is one of the principal causes of visual damage in diabetic population. And this progressive degeneration of the retina is believed to cause postural instability, both during quiet stance and during locomotion. I tell you here, this is a figure that the normality of the locomotion, that we have different phases, and the results of this instability is falling. Falling, especially in older age, is very problematic because it could lead to several damages, bone, joint, etc. So, because we know that the regular practice of physical exercise is beneficial in the management of diabetes, we sought to study the eye movement characteristics produced during the view of optical stimuli. Because, as I said, these optical stimuli stimulate our work in the environment. And we did it in two groups of diabetic people, active people and inactive people. So active people were 15 subjects who followed the 6-month exercise program 3 times a week. Inactive people were 25 diabetic sedentary subjects, so they just had sedentary behavior. So this is our setup. In the drawing here on the left, you see the screen with the dots, the representation of the visual stimuli you saw in the second slide. And our subjects were standing in front of the screen, fixating the fixation point, which was always in the middle of the screen. We used 3 types of stimuli, or at least we used more types of stimuli, but today I'm talking about these 3 stimuli. Full screen, we call it full screen because dots are in all parts of the screen. So the arrow indicates the direction of the moving dots. So stimuli were always expanding from the focus of expansion. Second, the stimulus was to stimulate just the peripheral visual field, so with the blank in the middle. And the third stimulus was to stimulate only the foveal part of the stimulus. Okay, so here you see a picture of one of our subjects. She's wearing the helmet. I will show you the helmet in the next slide. This is the eye tracker. She's in front of the screen, and this is a picture taken during the stimulation. So you see the peripheral stimulus with dots only in the peripheral part, and this is the foveal stimulus with dots just in the foveal part. For determining the exact dimension of the stimuli, we refer to studies presented in literature. So this is 7 degrees in radius, and this is 20 in radius. So we wanted to be sure to stimulate only the peripheral part and for only the foveal part.
Should you just come here because they cannot hear you very well?
Here.
Just like in the middle. Exactly, so they can.
Okay, sorry for shoulders in this case.
No,
okay. Okay, so this is the eye tracker. We use iLink to actually the companies from here, from this region in Canada. This is the helmet. The helmet is very, very light. It's 300 grams and has 2 mini cameras mounted here, and it works with an infrared light. So it's just a reflection of an infrared light. The subjects were 135 centimeters from the screen, and the visual stimuli were back projected on the screen. The 2 computers both recorded and delivered the stimuli. Our stimuli had a duration of 30 seconds, and we acquired 2 stimuli, 2 trials per stimulus. Data analysis. So during the data analysis, we usually discard data before and after 200 milliseconds of a blink or semi-blink in each trial. We identified microsaccades and saccades with the algorithm of Otero-Millen, and we calculated microsaccades rate, duration, amplitude, and peak velocity. You see here one of our main sequence of eye movements. You see amplitude, x-axis, and peak velocity on y-axis. This is important. This is very important because microsaccades are defined as small eye movements with an amplitude of less than 1 degree, so they have to be very tiny, and a peak velocity of no more than 100 degrees per second. All the rest are saccades. So you see what is shown in red is microsaccades in this range, amplitude and peak velocity, and all blue stuff are saccades. Everything bigger, much bigger, because those are detectable. In this analysis, because we wanted to see if the microsaccades can indicate the unconscious focus of attention, we used circular statistics. So basically, by studying, by studying the microsaccades angular orientation, we can have information about the focus of attention. In this analysis, we considered each vector of each microsaccades. So for each stimulus, we have about 4,000, 6,000 microsaccades. Microsaccades, in addition of being very, very tiny, are very, very rare. So they have a frequency of 2, maximum 3 microsaccades per second. So they occur very rarely. That's why I told you it's important to have an eye tracker with a high frequency of recordings, otherwise you don't detect them. And to analyze this data, we used Oriana, circular statistics, and we assessed the uniformity using the Rayleigh test. Okay, so those are results. You will see plots like this. I also I did something wrong. Okay, okay. Sorry, this is very nasty for me. I apologize for that, but it's very complicated. Okay, so I always put the stimulus here, so it's easy to follow the data discussion. So you see data in baseline here on the left, and after 6 months on the right, for both active and inactive groups. So again, active are people, diabetic people who follow the exercise program, and inactive are sedentary behavior. So for full stimulus, so the stimulus that covers the entire screen, we have significant mean vectors for microsaccade at baseline for both active and inactive. So you see the mean vector is indicated by the black line, and the red one is the circular standard deviation. So we have a significant trend toward, let's say, 100 degrees direction, so it's in front, slightly leftward direction for attention and conscious attention at baseline. Same thing for inactive, but as you see, inactive people tended at baseline to produce more microsaccades around the entire visual scene. So it looked like they were probably more curious than the others. After 6 months of exercise program, you see active people showed more or less the same trend than baseline. Inactive people was not significant. They reduced their microsaccades generation basically around these two axes. So that's why they moved a little bit on the right side. That was for full stimulus. Results for foveal stimulus baseline active were significant, while inactive were not significant. You see, it looks like they were very, very curious, and they tended to explore in a general way. So you see a uniform distribution of microsaccades here, while active people had a uniform and non-uniform distribution. Same thing after 6 months of training. Fovea active people were significant, so they had a common concrete mean vector for microsaccades, while inactive people did not show any significant value. Peripheral stimulus is more or less the same. They were both significant at the baseline, with inactive people exploring more in a diffuse way, but exploring more of the stimulus. They were all curious, probably, about the stimulation. After 6 months, only active people were steady, and inactive people reduced a lot their microsaccadic activity, being generally on the other side of mean vector. Those are the saccades, results for saccades. So we also studied the saccades. We didn't expect anything significant because those are different type of movements. I mean, saccades are eye movement that we use for exploring the visual field, and they don't indicate anything. They just drive our gaze to a specific spot in the visual field. We didn't find any significant effect of clustering in any situation. So this is a full stimulus. People made some saccade, of course. Inactive people always made more eye movements with respect to active people, but around they were distributed around the visual space, not being clustered anywhere. Same thing about phobia. You see the circular standard deviation here are very, very big for all cases. So there's no difference between active and inactive people in the generation of saccades. Same thing about periphery. I care to tell you something for saccades. Our subjects were instructed to fixate the central fixation point. So they, well, I'm not surprised about this lack of significance because they were instructed to fixate a point. So they were not instructed to explore the visual scene. That's why, I mean, I'll show you this data, but just to make you see the differences between saccades and microsaccades, basically giving our instruction, these movements are basically meaningless. So basically our active groups showed non-uniform or clustered, so if you want, distributions of microsaccades orientation in all visual stimuli. In both baseline and 6 months, the inactive group did not show any clustered attentional focus. No difference observed for saccade orientation. So these in the results could indicate that physical exercise modulates microsaccades activity, suggesting beneficial effects in heading perception processing. So I would like to spend 1 minute to explain you in detail what I mean, because people usually don't make connections between physical exercise, muscle, and muscles. So when we play physical exercise or physical activity or sport, whatever you want to call it, we have input signal descending from motor cortexes to spinal cord to muscles, and feedback signals from muscles through spinal cords to all possible areas in the brain. So this is a perfect way to keep healthy all these cells. So muscular cells work because we are asking the muscles to work together. The brain works because the brain needs to send signals and to elaborate the feedback signals from muscles and joints. And further, during physical activity, any kind of physical activity, we need to make integrations between signals from different modalities. And our brain needs to select a specific focus of attention. And if we play physical activity, or if we are active people anyway, we use these mechanisms to have a beneficial effect in the entire organism. So that's what people usually don't do, because physical exercise is something related to muscles and etc. While the brain is the most involved organ, because we activate muscles just because we have a brain. So our future direction is to analyze data from 18 months of physical activity program and 18 months and 4 months follow-up, because this is like longer projects we analyzed so far with baseline 6 months that I presented today, but we still work in progress. And the last thing to do for us is to analyze more stimuli, because we also use left heading with dots accelerated on the left, and right heading with the dots accelerated on the right, and two controls: random dots where there is no specific structured motion, and fixation in the dark with no stimulus. So I'll be very happy maybe next time to show you the entire sets of stimuli. Thank you very much for your attention, and I also wanted to acknowledge my university and the Ministry of the University and Research in Italy, because this is a project founded with a goal pre 2022 and it's also next generation. So thank you very much.
Thank you, Milena.
Thank you.
So maybe just take some questions.
Sure.
Anyone online has any questions, please don't hesitate to write your questions. Or you can unmute and ask the question directly.
Yes, of course.
I have a question. Very nice presentation, something very new for me. The field is really unknown for me. How many patients or you have per group? I don't remember the first one. It was
Right, well, this data refers to 15 active people and 25 sedentary people. But we enrolled a total of 79 people.
okay.
So slowly progressing because they didn't start at the same time. So right now, almost everybody finished a year of studies, so they are progressing. I mean, the active people are progressing. Then we keep recording every 6 months the other guys, the sedentary guys.
And this is regarding my question because the answer of this are probably heterogeneous. So you probably have to increase the number to have something that really reflect the
Absolutely.
I assume that if you take two diabetic patients, you will have two different answers.
Absolutely.
Yeah.
Absolutely. No, it's a work in progress because it's a sort of longitudinal study lasting almost 2 years. You know, it's very difficult to find subjects available for playing physical activity.
Yeah.
And it's a little bit easier to find the sedentary people, but they, you know, because all the other recordings takes about 1 hour and 20 minutes, even if it is every 6 months, is tough. So we're slowly progressing to increase the number to at least 35 and 35.
How do you divide them into those with physical activities on what basis?
Oh, their willingness to play physical activities. So if they are willing to follow the program and they assure us that they want to follow the program, they are included in the physical activity. Otherwise, they are included in the inactivity. Of course, we care not to have like somebody in between. You see, so a person who goes to have a walk, for example, sometime with friends, etc., is on the borderline of sedentary habits, you know, because this is better than nothing. So we care to have sedentary and active because we believe that physical activity is not able to restore previous visual functions if diabetic retinopathy is already present. But it could stop or it could delay in a very strong way the development of the problem. So if you are a person doing something, we don't know really in which group you should be better inserted. So we discard people who have some activity sometime. So we just want sedentary or follow our program.
I have a question
Sure.
about physical activity. So if people run outside, we can say the type is changing. But if some people do exercise in the gym, they just watch the empty wall. So another possibility is if I am just sitting in the living room and watch the virtual reality thing, VR. The size is changing, but I don't do exercise. Yeah, what happens for the microsaccades?
Okay, so the gym is actually a sort of halfway for exercising the brain, because you are perfectly right. If you play in a gym, you stay on a bike or on a treadmill. Well, on a treadmill also, but you stand on the bike, you know, you don't have a big optic flow. At the same time, you use your muscle. So you are something in between. So the gym is something very particular. But for example, when you have volleyball play or basketball, you are out, you are indoor, not outdoor, but still there are a lot of stimuli. So gym activity is more related to steadiness, even if we do cardio workout. I agree. But anyway, those feedbacks from the muscles are very much effective. So in this group of patients, we are not selective in the type of physical activity they play. They can go in the gym or swim or walk. But for us, it's fine with our team, but they need to practice 3 times a week. Our very future goal is to see if there is a specific indication for diabetes people in terms of which type of physical activity could be best for them. We don't know yet. I don't know if we will be able to answer sometime. But we divided these people in 3 sports, basically 3 types of exercises, and we will see if some data will arrive. I'm not sure because our sample is big because it's almost 80 people, but 80 people are not so many for a division in 3 groups. So we'll see.
So, sorry,
Not good.
repeated question. So if I don't like physical activity and if I watch the video, does it work?
No. Well, it works. Well, if you don't like physical activity for your brain, like crosswords or possible games helps. But watching TV is not helpful because you don't move. Watching video, virtual reality, it could be a good idea.
Yeah. But how about video games? People who are gaming all the time, do they take benefits of this?
I'm sorry, video games are very bad for the brain. I'm so sorry.
I'm not a video gamer.
I know, I perfectly know, but you know, this is what I try to explain to my kids as well. So video games are very bad for the brain because you are exposed to something artificial.
Yeah.
And you know, it's
In fact, they can increase the stress hormone.
Yeah.
exactly
Yeah.
first
Yeah.
thing. Second
Right.
thing, what they don't understand, because we are in an ophthalmology and visual science meeting, is that they play on the phone. When we are lucky, they play on tablet, which is a little bit.
Oh, yeah.
But it's small and very close by. So the reason why we develop myopia or possible, you know, problems is because we read too close and we stay too close
To the
to the surface. Further, these kids play in the dark or study in a dark environment, which is very bad for the eye because the eye needs light. So, you know, all this combination makes the eye doctor very happy, of course, but it's not working for the brain. I'm sorry. So we need to move.
The eye is, like, if you think eye as an isolated organ, then you can think of eye exercises. What we tell our patients is it is a part of the body. It's all integrated with everything else in the body. So
Indeed.
everything has to be in place if the eye has to be in place.
Exactly,
Yeah.
exactly, exactly.
And that's why maybe some gyms are having, you know, a nice view with big screens on the street.
Exactly, it's
or
good.
something like this.
It's good. It's good because you see the outside world. I understand.
It's less depressive.
It's less depressive. I mean, it's very good if people like the gym. Please go to the gym as much as you want. It's indoor, but if you like it, it's okay. So the main message has to be that each person has to find the proper way and the proper type of activity they like: dancing, gardening, volleyball, soccer, whatever.
Yeah.
But everybody needs to move to prevent diseases like diabetes.
Did you measure or observe other benefits than visual function to,
Yeah.
to cure the diabetes?
Absolutely. We also study, well, our setup is the one you saw. In addition, we record simultaneously electromyography and stabilometry. So we study the equilibrium and the motor activity. So what we saw so far in these people is that muscle activity is reduced in inactive people. And they have a bigger base of support, like that.
More
So
stable. They're more
they
stable.
are unstable.
Unstable.
Yes, they are more unstable. They cannot stay like that because there is weight. So if you enlarge the base of support, you are more stable. So when you see a larger base of support, it means the muscles are weak usually, or you need stronger muscle contraction to keep steadiness, because otherwise you oscillate back and forth, especially on the mediolateral domain. It's not like anterior-posterior, but mediolateral. And this is like propedeutic to be at risk of fall. It's like old people, very old people walk with a very large base of support because they are unstable, but physiologically. This is possible. This is a possible onset of diabetic neuropathy. That they don't feel any symptom yet because diabetic neuropathy is very nasty as well. But they're already neuronal damage, nerve damage, if they have a very large base of support. So far, we are studying these three dimensions besides the retina.
But in case of exercise, you will almost stable.
Exactly. Yeah, they show to be more stable. Yeah,
Okay.
that's benefit because in this case there is a fat reduction in the muscle, so the muscle is more performing and more active. So it's more important, and this is actually something that could be reversed. The muscle activity could be reversed. Neuronal activity and neuronal damages cannot, but muscle can compensate. So that's another important point.